Greetings in the name of the Lord Jesus Christ. I'm Salvador Cordova, and it is a privilege and honor to participate in this session on genetics, genomics, and information systems. My presentation today is entitled Fearfully and Wonderfully Made Memory Devices in Chromatin Complexes. My presentation will attempt to tie recent discoveries of memory devices in the DNA chromatin complexes to the theme of this year's CSC conference, namely, memory, tradition, and the future of faith. If you'll permit me, let me introduce myself and the organizations that I'm affiliated with. I'm a former scientist and engineer in the aerospace and defense industry with an educational background in physics, mathematics, computer, and electrical engineering, but have since migrated to the field of biology, where I serve as a research assistant for the FMS Foundation. I'm also a part-time visiting professor of Christian apologetics at Trinity College of the Bible and Seminary, a small international internet-based distance learning institution. My presentation today is made possible by the generous support of the FMS Foundation. The FMS Foundation was created by Dr. John Sanford, who is a renowned geneticist and inventor of 40 years at Cornell University. His invention, the gene gun, is part of the collection of the Smithsonian National Museum of American History, because at one time a sizable proportion of all the genetically modified organisms on planet Earth were created through his gene gun. John also published a landmark book, Genetic Entropy, which explores the effects of mutations on the deteriorating human genome. The photos shown, going from left to right, begin with a picture of John with his gene gun in the mid-1980s. The next photo is the gene gun on display at the Smithsonian National Museum of American History. Next is a picture of John from the back cover of the 2014 edition of his book, Genetic Entropy. Some of what I present today is research related to supporting the thesis of John's book. John converted to Christianity early in his scientific career, and as a result has spent the better part of the last 2 decades declaring the works of God in nature, particularly in biological systems. I serve as John's research assistant and on-site correspondent at the National Institutes of Health, where I report on discoveries in the field of epigenetics, epitranscriptomics, and higher-order chromatin structures, fields on which I briefly touch upon today. I must, however, point out the theological views I express here today are ultimately my own views and not necessarily anyone else's. The key point I hope to cover today is Psalm 139:14, which says, I praise you, for I am fearfully and wonderfully made. Wonderful are your works. My soul knows it very well. Let's start with some basics which I hope many of you are familiar with. The first artificial memories were made of clay tablets, parchments, etc. Modern artificial memories are electronic and chemical. There are a variety of artificial memories such as compact discs, disk drives, etc., but also things such as computer read-only memory, ROM, and computer random access memory, RAM. Information flow and/or memory is measured in bits and bytes. There are the familiar terms like bits, kilobits, or bytes, kilobytes, etc. We can apply information metrics to describe the memory and information processing capabilities of man-made computers, but we can also apply information metrics to God-made computers, such as this God-made computer depicted here, known as the human cell. Some people may object to me calling a cell a God-made computer. First, if one prefers the term nature-made over God-made, that's fine. But as a figure of speech and for the purposes of this talk, I will call the cell a God-made computer. Second, there is strong sentiment by researchers in relevant fields that the cell is indeed a computer. For example, that was the explicit thesis in the chapter entitled Living Cell as a Universal Computer in the book Cybernetics and Systems Analysis, published by Springer in 2013. And also the sentiment of genetic engineers like Craig Ventner, who said life is a DNA software system. And last but not least, Bill Gates said, DNA is like a computer program, but far, far more advanced than any software ever created. If we were to compare God-made computers with man-made computers, we see that God-made computers are far more sophisticated. God-made computers are physically self-healing from damage, are self-manufacturing, are self-assembling. For those familiar with computer science terms, God-made computers implement von Neumann, self-replicating automata, and Quine algorithms far more complex than those found in the man-made world. These cellular computers affirm Psalm 139, that we are fearfully and wonderfully made. What I depicted earlier was a generic kind of cell, a somatic cell, but there are specialized variants of the basic generic cell in humans, such as the nerve cell. The generic cell is on the left, the specialized nerve cell or neuron is on the right. Now, in the man-made world, we can connect man-made computers with devices like a network switch and other sorts of networking equipment to form a variety of networks like local area networks, wireless networks, the internet, etc. And by the way, here's a conceptual picture of the internet. But not only can networks be made by connecting man-made computers, networks can also be made by connecting God-made computers called neurons to create a brain. Let me show a video. Here is a nerve cell, also known as a neuron, receiving and transmitting electrochemical signals from other neurons. Neurons usually receive signals through wires called dendrites and transmit signals through wires called axons. Each neuron has about 1,000 such connection wires to other neurons, and there are about 100 billion neurons in the human brain. The process of thinking, imagining, feeling, remembering, learning, hearing, seeing, talking, tasting, smelling, creates patterns of information exchange, storage, and processing by neurons. Although we barely know any of the details of how this information exchange and processing actually works, we do know in general the power to reminisce, to dream, to conceive, to be inspired, to recognize a familiar face, to recognize a familiar object. The power to do abstract reasoning, to do science, is driven by the networked exchange and processing of information between these God-made computers called neurons. Let me summarize a present-day estimate of the brain's memory capacity. Memory and information are measured in bits and bytes as mentioned earlier. Network size is measured in terms of the number of devices connected to the network, the number of connections between the devices, and the number of switches involved in those connections. The brain's memory capacity is on the order of 2.5 times 10 to the 15th bytes, according to an article in Scientific American 2010. And from an article in Science Daily 2010, we have this passage. "The brain's overall complexity is almost beyond belief," said Dr. Stephen Smith. "One synapse by itself is more like a microprocessor with both memory storage and information processing elements than a mere on-off switch. In fact, one synapse may contain on the order of 1,000 molecular scale switches. A single human brain has more switches than all the computers and routers and internet connections on the earth." Maybe it's worth repeating the sentence I bolded. A single human brain has more switches than all the computers and routers and internet connections on earth. This again affirms the claim of Psalm 139, "We are fearfully and wonderfully made." I just talked about the collective networked memories of God-made computers called neurons or nerve cells. What about the memories of the individual cell? Well, cells have their own individual memories. Chromatin is but one of several memory systems of the cell. I will describe chromatin shortly. Chromatin implements both ROM and RAM memory systems simultaneously. That claim is not only mine, but also found in the Stem Cells Handbook 2013. There are other memory systems in the cell which I cannot cover today, but I mention them for completeness. These are systems such as the transcriptome, epitranscriptome, phosphoproteome, and glycome, etc. It's worth mentioning the glycome is probably the biggest information system of them all, dwarfing the chromatin DNA system, but also the least understood, so I can't possibly cover it today. But anyway, going back to the generic cell, not just the nerve cell, let's explore what chromatin is. First, I'll circle in red the cell's nucleus, and then I'm going to zoom in on the nucleus with another diagram. This diagram is of the nucleus and the chromatin contained in the nucleus. If we conceptually pulled the chromatin out of the nucleus as is done in the diagram, you can see first the chromatin fiber, which is a compacted form of chromatin. Chromatin is basically what looks like beads on a string, which I just circled in red in the diagram. This is a conceptual diagram, but I'll show you a corresponding actual electron microscope photograph of chromatin. You can see here the scale at 0.05 microns, which is also 50 nanometers. So we see this God-made bead on a string pattern for a memory system in the cell. But looking at this, it reminded me of the early man-made computer magnetic memory systems, RAM memory systems, pictured here, albeit deliberately made to look grainy, like the grainy quality of an electron microscope photograph. In fact, if we go back even farther in man's history before the computer era, we see the beads on a string architecture for memory on the first mechanical calculator called an abacus. The ancient Inca abacus is on the left, and the Chinese abacus is on the right. The resemblance between God-made and man-made memory systems is purely superficial, but I found the resemblance quite amusing nonetheless. So going back to the details of chromatin with the beads on a string architecture, I'm going to circle in red a memory bead. The memory bead is called a nucleosome. The nucleosome contains about 40 bits of random access memory. There are about 16.5 million such nucleosome beads in each cell. Let me zoom in on the bead, the nucleosome, with another diagram. This blue diagram is a zoomed-in view of the nucleosome. And we see within the nucleosome are these things called histones. The histones have those red tails in the diagram. The tails are made of amino acids. These tails have places to read and write random access memory in the form of chemical modifications on individual amino acids on the histone tails. There are a host of chemical machines to read, write, and erase the memory marks on histone tails. As said earlier, the sum total of memory for all the histone tails in a single nucleosome is about 40 bits. This system of RAM memory is better known as the epigenome. The field of study of the epigenome is known as epigenomics or epigenetics. The terms epigenomic or epigenetic are fairly interchangeable except for a few subtle situations. So now I'm going to zoom out. Summarizing again, the memory bead is called a nucleosome. If we take into account the fact there are 40 bits of RAM per nucleosome, 16.5 million nucleosomes per cell, 100 trillion cells per human, 1 byte per 8 bits, a back-of-the-envelope calculation yields a figure of 8.3 times 10 to the 21st bytes of network epigenetic RAM for an adult human. That's a lot of memory. So how is this epigenetic RAM used? Well, it's used partly to provide memory for neurons, which provides memory for the brain. This was a surprising discovery in neuroscience and is discussed in science journals such as Nature Neuroscience. Epigenetic memory, in addition to helping with brain function, is also used for development of the body from fetus to adult, and in the self-healing of our bodies from injury, because the epigenome is involved in development and differentiation of stem cells. Now going back to the chromatin diagram, I'll focus on the string part of chromatin, which I circle in red and which should be familiar to most of you. The string part of chromatin is DNA. DNA is a read-only memory string. DNA is composed of chemical base pairs such as the one I'm highlighting. Chemical base pairs implement the alphabet of DNA. The alphabet of the DNA language is composed of only 4 letters, A, C, G, and T. The director of the Human Genome Project and the National Institutes of Health, Francis Collins, said at a White House ceremony, "DNA is the language of God." If so, A, C, G, and T is the alphabet of the language of God. Each base pair contains 2 bits of ROM. There are about 3.3 billion base pairs per human cell that result in a total of 6.2 gigabits or 786 megabytes of genetic read-only memory, ROM. I'm going to zoom in on the DNA a little more and show just one more memory system on the chromatin. So this is a zoomed in view of the DNA part of chromatin with the familiar DNA double helix. What can be done to the DNA is attach methyl group molecules that serve like little post-it sticky notes on the DNA. This post-it sticky note adds a little more epigenetic RAM capability to the chromatin. There are about 28 million methylatable sites, that is, places to post these sticky notes on the DNA, in each human cell for a total of about 27 megabits or 3.3 megabytes of additional epigenetic RAM memory per cell. In summary, a single human brain has more switches than all the computers and routers and internet connections on Earth. The human body is a network of 100 trillion God-made computers with a sum total of 8.3 times 10 to the 21st bytes of epigenetic chromatin-based RAM. Epigenetic RAM is used for brain function, body development, and healing through stem cell differentiation. My closing remarks will focus on a long-standing paradox, the paradox of a simultaneously wonderful yet miserable world. On the one hand, we have fearfully and wonderfully made minds and bodies that live in a universe miraculously fine-tuned for life, for scientific discovery, and for comprehensibility. But on the other hand, our fearfully and wonderfully made bodies will suffer and die of old age, and our fine-tuned universe will also suffer and die through the second law of thermodynamics. The 1979 Nobel Prize-winning physicist Steven Weinberg, as he pondered the paradox of a universe wonderfully structured for life and for scientific discovery, yet simultaneously a universe ordained to die, said, "The more the universe seems comprehensible, the more it also seems pointless." He could not come to terms with the fact this present universe was subject to futility despite looking purposely designed to be comprehended by our human minds. Furthermore, our minds and bodies are so complex and therefore fragile, and the number of birth-defected mutations relative to our reproductive rates is so high, that based on the work of 1946 Nobel Prize winner Hermann Muller, many modern-day geneticists have predicted the human race is evolving toward genetic deterioration and extinction rather than toward improvement. The geneticists who hold this view include John Sanford at Cornell, Brian Sykes at Oxford, Michael Lynch at the National Academy of Sciences, and many other geneticists. That prediction is controversial, to be sure, but the prediction is testable, and time will bear out if it is correct. Unfortunately, the question of genetic deterioration, or genetic entropy, to use John Sanford's term, is ultimately moot, because what is not controversial is that the solar system and universe will eventually die as ordained by the laws of thermodynamics, thus fulfilling the words of Jesus, who prophesied, "Heaven and earth shall pass away." In consideration of these ideas, one may ask, would it not have been better if God, the great playwright in the sky, just created a perfect world to begin with, and thus bypassed suffering and pain, and the problem of having to recreate everything all over again, with a new heaven and a new earth? My answer is, not necessarily. Because the best happy endings in stories must involve some suffering in the early chapters of the story. There is no great drama or story or happy ending without some pain in the early chapters. God is the great playwright in the sky who writes the divine drama where the present suffering of this age is building for us an eternal weight of glory beyond all comparison, just as the Apostle Paul taught us in his second letter to the Corinthians. That explains in part why God might create a universe that self-destructs, in order that it gives special meaning to the new heavens and new earth. By the way, while I'm on the topic of self-destructing designs, there is the curious fact that Monsanto genetically engineered self-destructing seeds which farmers can plant and harvest for food, but which create sterile crops that can't be used as seeds to plant for the next season. This forces farmers to go back to Monsanto every year to buy new seeds. These self-destructing seeds are permanent reminders to the farmers of their need of Monsanto. By way of imperfect analogy, our self-destructing universe is a sober reminder that we humans are not ultimately self-sufficient, but are in need of a savior. The difference, however, between Monsanto and God is that Monsanto builds self-destructing designs for their profit, whereas God builds self-destructing designs like our universe for our profit to build an eternal weight of glory in the next life. The Apostle Paul, in his letter to the Romans, understood that God designed the universe to self-destruct in order to give meaning to the new heavens and new earth, thus resolving Weinberg's paradox of a wonderful yet apparently futile universe. In closing, I quote the great physicist Lord Kelvin, as he reflected upon the laws of thermodynamics which he himself helped pioneer. He said, "We have the sober scientific certainty that the heavens and earth shall wax old as doth the garment. Dark indeed would be the prospects of the human race if unilluminated by that light which reveals new heavens and a new earth."